both my title and acknowledgement slide. I'm a postdoc with Yaniv Brandvain, and these people, particularly Jill Ecker and Natasha Sherman, have been huge in getting this data together, and this work is supported by a hatch grant to Yaniv. So I'm going to talk today about a population of wild sunflowers that's been historically described as a hybrid swarm. So why are we interested in this thing? Often, I don't want to say usually, often when we're interested in hybrid populations, it's not the hybrids per se that we're interested in, but what they say about the process or result of speciation. And so when you get individuals in secondary contact after divergence, other populations or species, you start mixing up those genomes, you get lots of different genotypic combinations, and that might be able to tell you something interesting about what genes underlie, say, intrinsic incompatibilities between species or interesting phenotypes that might be adaptive in different environments, but in the hybrids, these genotypic combinations don't show up. So that's kind of the why, but I'm getting ahead of myself a little bit with all that. What we're going to look at here is, I'll call it an old hybrid swarm. We don't know how old. It was first described in 1950 on my birth date of all days, so I feel very personally connected to it. It's between these two species, Helianthus divaricatus and Helianthus microcephalus. This is down near Bloomington, Indiana. So those are two sister species. I don't quite know exactly how diverged they are or when that split time was, but we do know that they have hybridized. There's a species called Helianthus glaucus that we now know is actually representative of a hybrid between the two. We've also looked at some of the USDA germplasm. We know that these things do seem to form hybrids, but you'll see soon what's going on in our population. So Helianthus divaricatus is a plant that has relatively large heads for a sunflower. It lives kind of in these drier areas, more open. Flowers a little earlier in the year. Helianthus microcephalus has small heads, as you might be able to grok from the species name there. It lives in maybe more shady or wetter sites, flowers later in the year. But they do form these hybrids. These hybrids typically exist in populations that are intermediate with respect to moisture, things like that. Sort of a traditional occurrence for hybrids. And so, what's my next slide here? Like I said, we do have historical evidence that these things are forming hybrids and intergrades of all sorts, back crosses. So these are histograms of hybrid index, and so on the 0 side of this, you've got pure Helianthus microcephalus. On the 10 side, you have pure Helianthus divaricatus. And these are 5 different characters that are scored on a scale from 0 to 2. And so if you're intermediate, you get a 1, and you just add all that up, and you can get anywhere from 0 to 10. So there is the first description in 1950. We see maybe a lot of microcephalus, but things that span the phenotypic range. 11 years later, in 61, we see the composition maybe changing a little bit, and then 11 years after that, in 1972, we get some maybe shift back to microcephalus, but this thing looks dynamic, and there's interesting stuff happening phenotypically here. So our contribution is going to ultimately be to the genetic side of this. But what Yaniv and Natasha did was go down to Indiana, sample about 200 individuals from that hybrid population. And they sampled about 8 individuals each of the pure species at locations removed from each other and the hybrid zone. So there's a picture of renowned field biologist Yaniv Brandvain collecting here. So this is, I forgot to mention, that the hybrid zone we're looking at is in a power line cup. So Yaniv and Natasha just ran a transect down 100 meters of that thing and collected plants off either side of it and took precise measurements about where they collected those things. So it's really cool because we have a really detailed map of where individuals occur in that zone. So each little green dot here represents a plant that's either sort of along and off the transect. So like I'm saying, we know where these plants are. We know something about them. So we brought them back. These things are perennial through tuber formation, so you can go in and sort of pull out a little bit of tuber and grow it up in a greenhouse without destroying the plant in the field. And so we did that. brought them back, potted them up, grew them in a common garden, if you will, in a greenhouse in Minnesota, and we took some first-pass phenotype measurements. And so these bars, the colored bars, correspond to the different species, and those are ranges that were used in scoring those phenotypes in the previous work. So anything with a disc diameter, and that's that little circle there, under this range would have been scored as Helianthus diverricatus and microcephalus. And so there's disc diameter, and we also did it for petiole length, which is like kind of a little branch that attaches the leaf to the main stem. And so we have an expectation here. These are two different. There's not an overlap apparently in the species, so let's see what we find when we collect those phenotypes. And what we see is some bimodal distributions here. On the disc diameter, that falls nicely within the range of Helianthus microcephalus. We've definitely got a peak here. It's not under here, but who knows what's going on there. I mean, that's maybe an effect of the environment it's grown in. Helianthus diverricatus doesn't have a petiole really. It's just kind of sessile. It's attached right to the stem there, so we see a lot of zeros there and sort of this peak and petiole length within Helianthus microcephalus. So that's the phenotypic description. It's maybe not all that encouraging that we're dealing with a hybrid swarm, but our main contribution, like I said, is going to be on the genetic side of this. So we did a bunch of genotyping. We used RADseq. RADseq's great for looking at population structure type stuff. RADseq, I don't know how much I need to explain it, but it's reduced representation sequencing. We get a lot of sequence all across the genome. We process that in stacks, do a lot of standard bioinformatics fun, filtering for quality and everything, and we get, importantly, two different kinds of data that we're going to use here. One is SNPs, which we'll use for like clustering algorithms like structure admixture, and the other is haplotype. So the nice thing about RAD is that you get maybe 95 base pairs, but it's all perfectly linked. You know what the haplotype is at your RAD locus. So I kind of just jumped right into it. When we take those SNPs and we run it through admixture, this is what we get. So down here, this is the individuals from the putatively pure Helianthus microcephalus and Helianthus diverricatus populations. We see 5 admixed individuals maybe, and a lot of things that look like pure species. Annoyingly, in our pure population, we have an individual that looks pretty admixed, and we see some signal down there. So what if we just map that on to those plants that we have? We see that these species are right on top of each other in the field. But boy, oh boy, I don't know what to say about this. I mean, I'm not sure you can glean much from just looking at this pattern, but, you know, we see these things cropping up, and they're between both species, and cool. I would have rather had a lot more purple triangles to do cool stuff that I mentioned in my intro, but this is the reality. There's something cool happening for sure. But that's just cluster membership. That's just saying, okay, I want 2 populations, check out the allele frequencies, and push stuff as a mixture or, you know, some version of that. And we can learn actually a fair bit more about co-ancestry if we look at those haplotypes. So we used those haplotypes and did something called RAD painting. This is an extension of something you might have seen in the human literature, primarily for painting chromosomes. It does a lot of smart things with HMMs and everything, but you can also just employ it here because you have haplotype data. So we paint a haplotype for an individual. So for each individual, for each RAD locus, you can go through and ask, who's my nearest neighbor? And it's agnostic to what the actual species is, but you can say, just based on how many SNPs are different between all these haplotypes, who's my nearest neighbor? And that individual is someone who you share recent co-ancestry with. So again, you do that across the genome for every individual. You group that up. There's this MCMC clustering algorithm called Fine RAD structure that takes that information about your co-ancestry and puts all these different groups together. And so what you get out of it is a co-ancestry matrix, which is pretty cool to look at. I don't know how well that comes through here, but every row here is an individual, and this represents a scale where you share very little co-ancestry to more, basically. We'll think about it that way. And so what we see here is, you know, we're kind of recapitulating our structure result, which is we get a lot of individuals clustered together that probably are that microcephalus species, and this is Helianthus diverricatus down there. But we do get those hybrids back, and what's really cool about this is that when you start to look closely, those hybrids, you can start to see probably where or who their closest relatives are in either species. So that little red line for this individual probably corresponds to those individuals have some degree of relationship with that hybrid. And the same thing over here. So this right here is that individual that was hybrid in the pure species. And sure enough, it doesn't show here, but that's the other pure microcephalus species. So these things are admixing even when they're not that close, which is kind of funny because the plants in our population are right on top of each other and are doing it, or not doing it. And so this is a little bit tangential to what our aims were here, which was just to characterize population structure, but or to get a sense of how hybrid this population actually was. But this is a zoom in on the variegatus portion, and you can start to see where probably family structures within that population. And so sort of the darker you are, the more co-ancestry you share with those individuals. And so this might be a cool thing to look into in the organization of families on that very well-described population we have. And so to wrap it up, I guess I'm speeding through this. I thought I'd hear a bird by now. Something cool is happening out there. I don't know what it is. It's maybe not what we thought we were going to see when we looked at it, but some kind of cool, something's happening. I don't know what it is, honestly. We have these tubers in a field in Minnesota, and so when they start to flower this year, we can check to see, now that we know to which species each of these individuals belongs, we can go say, okay, do these individuals just flower at different times? There it is. And start to get a sense there. We can also try to generate some artificial hybrids and maybe try to map any incompatibilities that are there. But that's all work that will happen now. And with that, yeah, that's the end. All right, thanks everybody.